Hi, my name is Matt Tyska. I'm an associate professor in the Department of Cell and Developmental Biology here at Vanderbilt University Medical Center. And for the next few minutes, I'd like to tell you a little bit about our most recent discovery on the mechanism of brush border assembly. So the context here is the small intestine, specifically the transporting epithelial cells that line the small intestine. These are known as enterocytes, and on their apical or luminal surface, they build an organelle known as the brush border. Now, the brush border is the organelle that's responsible for nutrient absorption. So the brush border is essentially an array of membrane protrusions that extend off the apical surface of the cell into the intestinal lumen. Now, these protrusions are known as microvilli, and they're supported by bundles of actin. Now, collectively, these microvilli serve to scaffold apical membrane, and such that if an enterocyte builds more microvilli, it's capable of storing more apical membrane with its associated transporters and channels on the apical surface for function and nutrient absorption. So the brush border has several interesting morphological features that biologists have been trying to understand for many, many years. The first one is the tight packing of microvilli. These structures have very little free space between them, and when the brush border is viewed en face or in cross-section, we see this striking hexagonal packing pattern, which really suggests that the packing of these protrusions has been optimized to maximize their number. The second feature of interest is the uniform length of these protrusions. All the microvilli in a single brush border are essentially the same length, plus or minus a few percent, and the length control mechanism governing this endpoint has not been clearly defined. So the questions that we are trying to answer with the current study are: how do enterocytes build the brush border, and how do they achieve this exquisite level of organization that's really a defining feature of the brush border in its fully differentiated state? So this study started several years ago when we were looking at scanning electron micrographs of the apical surface of differentiating epithelial cells. These are cultured cells. And what we found is that microvilli coming off the apical surface early in differentiation have a tendency to stick together. So we became interested in pursuing the molecular basis of this observed microvilli clustering because we figured if there was some adhesion inherent to the surface of microvilli, this could solve the tight packing problem from a time point very early in differentiation, and it could also drive the overall process of brush border assembly toward that optimized endpoint, that hexagonal packing pattern that we see in cross-section. So when we took a closer look at our cell culture model and also at native tissue using deep etch electron microscopy, what we saw is that there are small thread-like links physically connecting adjacent microvilli in these clusters. So we got a clue as to the molecules that might be playing a role in intermicrovillar adhesion from a previously completed proteomic analysis of isolated brush borders. And that data set told us there were two protocadherin isoforms in this system that were poorly characterized in terms of their function. One of them was protocadherin 24, the other one was mucin-like protocadherin. Both of these are members of the cadherin superfamily of calcium-dependent extracellular adhesion molecules. So when we started our analysis of these molecules, we quickly saw some interesting things. First, both protocadherin 24 and mucin-like protocadherin demonstrate a striking localization at the tips of microvilli. They're highly enriched at the tips. And this is the site where we see these thread-like links forming between these protrusions. So clearly both protocadherins are in the right place to function in the formation of these structures. The second thing we found is that knockdown of protocadherin 24 or mucin-like protocadherin had a profound impact on brush border assembly in the context of an intestinal epithelial cell culture model. In both cases, what we found were brush borders that were very poorly organized. Microvilli were highly variable in length, and the tight packing that's characteristic of this organelle was completely absent in this system. Now, the third thing we found using an in vitro biochemical approach is that both protocadherin 24 and mucin-like protocadherin are in fact bona fide adhesion molecules, but they prefer to bind to each other in a heterophilic complex. We're able to reconstitute adhesion using bead aggregation assays, and we could also find, using scanning electron microscopy of aggregated beads, that thread-like links comparable to what we see on the surface of cultured epithelial cells or in tissue were formed in these reconstituted systems. Next, we became interested in trying to define the mechanism that drove the localization of protocadherin 24 and mucin-like protocadherin to the tips of microvilli. And for this, we used a combination of yeast-2 hybrid and biochemical pull-downs to search for cytoplasmic binding partners. We found two molecules, harmonin, which contains 3 PDZ domains, and myosin 7b, an actin-based motif that has a very complex cargo-binding tail domain. Both hormonin and myosin 7b target specifically the tips of microvilli, just like the microvillar protocadherins. Now back to hormonin for a second. This is an interesting molecule because of its direct connections to human disease. Mutations in hormonin have been linked to type 1C Usher syndrome, which is characterized by deafness and blindness. Now patients with type 1C Usher syndrome also present with enteropathy, although the cellular basis for these symptoms and, in general, the function of hormonin in the intestinal tract have yet to be studied. Now, to determine if intramicrovillar adhesion and, in turn, brush border assembly depend on hormonin and myosin 7b, we turn to the hormonin knockout mouse, set out to characterize the intestinal phenotype of these animals. Interestingly, we found that myosin 7b and mucin-like protocadherin failed to localize in these animals. They were no longer found at the tips of microvilli, and the consequence was a significant disruption of brush border organization. In the small intestine and the colon, we found cells that were unable to build normal brush borders with well-packed microvilli of uniform length. Now, in combination, these data lead us to propose the following new model for brush border assembly. In differentiating enterocytes, protocadherin 24 and mucin-like protocadherin target to the tips of microvilli by virtue of their interactions with hormonin and myosin 7b. Now, once at the tips, these protocadherins can form heterophilic adhesion complexes between adjacent protrusions, and this drives the clustering of microvilli. Now, as differentiation proceeds, microvillar clusters get larger and larger, incorporating more and more protrusions, and eventually the cell is left with a single large-scale cluster on its surface, a fully differentiated brush border. Now, these findings also have two other important implications. First, they likely provide a cellular and molecular basis for the GI symptoms reported in type 1C Usher syndrome patients. Second, they suggest that extracellular adhesion is one mechanism that cells might use to control their shape, and this is one general concept that we're going to test moving forward with future studies.